up to special tissues. This is up to special tissues. So, next part is up to special tissues. Next part is up to special tissues. So, we have to discuss here two parts. Completed up to simple tissues. Now, complex tissues and special tissues. Just now I told you, complex tissues means group of cells with dissimilar structures. Group of cells with dissimilar structures, but showing a common function. These are popularly called vascular tissues or conducting tissues. These are of two types, xylem and phloem. Special tissues are polyploid in nature because they are formed by the fusion of the cells. Polyploid in nature. Special tissues are formed by the grouping of cells based on their position, based on their secretion, based on the place where the secreted substances are stored. Based on the place where the secreted substances are stored, these are divided into different types. Just now I told all the names: digestive glands, nectary glands, osmophores, secretory cavities, then hydatodes, latissiferous tissues and other things. Now coming to the first question. Arrange the following of hydatode in a correct sequence from inner to outer region. Hydatode is a special tissue that helps in the special concept called guttation. Guttation means loss of water in the form of water droplets from aerial parts of the plant body. Every hydatode structure will be like this. Every hydatode shows structure like this. Permanently opened or non-functional guard cell. Non-functional guard cell. Loosely arranged parenchymatous tissue called epitheme. This is an important bit. Loosely arranged parenchymatous tissue called epitheme. A special water cavity or water chamber. Then blindly ended vein endings or blindly ended xylem vessels. This is the structure of a hydatode. The xylem endings store the water in water cavity. Because it is a non-functional guard cell, water is released out in the form of water droplets. The guttation is directly proportional to root pressure. Root pressure means a positive hydrostatic pressure developed in the roots due to the accumulation of absorbed water. Positive hydrostatic pressure developed in the roots due to the accumulation of absorbed water is called as root pressure. The guttation is directly proportional to root pressure. So, whenever root pressure is more, guttation is more. Whenever root pressure is less, guttation is less. And the answer here is first option 3, 1, 4, 2. That is first vein ending. This is from inner to outer side. First vein ending. Then epitheme. Above the vein ending or vein ending is enclosed in the parenchyma. Then 4, air cavity. Then water stomata. Water stomata means permanently opened or non-functional guard cells. This is the arrange the following of hydatode in a correct sequence from inner to outer region. Next. The cells that originate from common mother cells in phloem. The cells that originate from common mother cells in phloem. Phloem normally contains the cells like sieve cells, sieve tubes, companion cells, sieve cells, sieve tubes, companion cells, phloem parenchyma and phloem fibers. These are the special components present in any phloem. Sieve cells are found in pteridophytes and gymnosperms. They are long columnar cells with nucleus initially, but nucleus is absent later. Sieve tubes are well organized structures in angiosperms. In sieve cells, you will find unspecialized sieve areas without sieve plates and sieve pores. In sieve tubes, you find specialized sieve areas with sieve plates and sieve pores. Companion cell is a special parenchyma cell which is very closely associated with sieve tube. The sieve tubes and companion cells both originate from a same parental cell. That's why these two are called sister cells. Companion cells and sieve tubes always originate from same parental cell. That's why they are called as sister cells. Companion cells and sieve tube difference is sieve tubes also lose nucleus at maturity. That means it becomes enucleate. But companion cells retain nucleus even at maturity. So, the cells that originate from common mother cells in phloem. Common mother cell means common meristematic cell in phloem is sieve tube elements and companion cells, not phloem fibers and phloem parenchyma. Companion cells and phloem parenchyma, sieve tube elements and phloem fibers. Answer is sieve tube elements and companion cells. These originate from the common parental cell. That's why both are popularly known as sister cells in plants. Tracheids are found in tracheids. Xylem is divided into two parts. Xylem is divided into four parts. Xylem tracheids, xylem trachea or vessels, xylem fibers and xylem parenchyma. These are the four parts in this. Xylem tracheids are found in lower group of plants like pteridophytes and gymnosperms, and also they are found in angiosperms but associated with xylem trachea. These are found only in angiosperms. Xylem fibers are two types. Librifa fibers, two fibers with simple pits. Fiber tracheids, non-functional xylem tracheids converts into fibers with bordered pits. The only living tissue in xylem is xylem parenchyma. So, now the question is xylem tracheids are found in. Xylem tracheids are primitive type of xylem found in lower group of plants like pteridophytes and angiosperms, pteridophytes and gymnosperms. They are also found in angiosperms but associated with xylem vessels. Number of xylem vessels will be more and number of xylem tracheids will be less. So, xylem tracheids are found in pteridophytes, gymnosperms and angiosperms. Answer is 3. Answer is 4. 1, 2, 3. This is a matching regarding special tissue. Tissue example function. Osmophores present in orchids. Osmophores present in orchids producing nectar is wrong. It does not produce nectar, it produces a pleasant smell. So, osmophores present in orchids producing nectar is wrong. Nectar is passiflora attract insects. In passiflora, you will find extrafloral nectaries. Nectary glands are present on the petioles in passiflora, and it attracting insect is correct because always nectar is a sugary sharp which attracts the insects and help in pollination. Simple laticifers carica secrete latex. Simple laticifers secrete latex is correct, but carica is wrong. Simple laticifers are found in nerium and some plants, not in carica. It is found in nerium, hibiscus, something like. Lysogenous cavity citrus producing oil. Lysogenous cavity citrus producing oil is also wrong. The correct combination is only two, that is nectaris, passiflora attracting insects. In citrus, it does not produce oil. It is a lysogenous cavity which stores an oil which is said to be volatile. That means once it gets exposed, it gets evaporated. So, only 2 is correct. It is an assertion. Ray parenchyma is a secondary permanent tissue. Ray parenchyma is a secondary permanent tissue is correct because ray parenchyma formed during intrastellar secondary growth. It is a secondary permanent tissue. It is formed from cork cambium is wrong. Why? Because in plants, vascular cambium or cork cambium contain 2 types of cells. One is fusiform initials, the other is ray initials. Fusiform initials and ray initials. Fusiform initials are spindle-shaped cells loosely arranged and they divide and redivide to produce 1. Secondary xylem, 2. Secondary phloem. Secondary xylem and secondary phloem. Ray initials are special circular cells very compactly arranged. They divide and produce rays. The rays are of two types. Ray formed in xylem is xylem ray or wood ray. Ray formed in phloem is phloem ray or bast ray. And ray formed in medulla called secondary medullary ray. So rays are formed not from cork cambium. Rays are formed from special cells in the vascular cambium or cambial ring, which is popularly called ray initials. So ray initials gives rise to secondary permanent tissues called ray parenchyma. So assertion ray parenchyma is a secondary permanent tissue is correct, but it is formed from cork cambium is wrong. It is not formed from cork cambium. It is formed from vascular cambium. So A is correct, R is true. Unspecialized sieve areas, sieve plates, perforation plates, bordered pits, advanced trachary elements, non-functional primitive trachary elements, primitive sieve elements, fiber tracheids, advanced sieve elements. A is 3. The answer is 4. A is 3. Unspecialized sieve areas, primary sieve elements which are called sieve cells. Sieve plates. Sieve plates is primitive sieve elements. Primitive sieve elements. Answer is 4. Sieve plates is advanced sieve elements. Fifth option. Fifth option. Then perforation plates. Advanced trachary elements definitely shows perforation plates. Why? Because in trachary elements like xylem vessels, the upper portion, the cells where the xylem are fused, the end walls are open. Near the open end walls, you will find special plates called perforation plate. So advanced trachary elements is perforation plates. Bordered pits. Bordered pits is non-functional primitive trachary elements. Non-functional primitive trachary elements means fiber tracheid. So second one. Fourth one is the answer. Vesselless angiosperms. Normally xylem vessels means they have to be present in angiosperms, but there are two vesselless angiosperms. The vesselless angiosperms are Dremis and Trochodendron. The vesselless angiosperms are Dremis and Trochodendron. Normally vessels must be absent in pteridophytes, but vessels with or vessels are seen in pteridophytes like Selaginella and Equisetum. Selaginella and Equisetum shows vessels. Then vessels are normally absent in gymnosperms, but a gymnosperm shows vessels, that is Neetum. So vessels must be present in angiosperms, but it is an advanced character found only in angiosperms, but there are some angiosperms without xylem vessels, Dremis and Trochodendron. Vessels must be absent in pteridophytes, but there are some pteridophytes with xylem vessels, Selaginella and Equisetum. Vessels are normally absent in gymnosperms, but there is one gymnosperm with xylem vessels, that is Neetum. So vesselless angiosperms are Dremis and Trochodendron. Answer is 3. This is an assertion reasoning question. Each sieve element usually associated with one companion cell. Each sieve element is usually combined with one companion cell. Sieve tube elements and companion cells originate from a common meristematic cell. Each sieve element is not associated with one companion cell. Every sieve tube has its own companion cell. That means sieve element contains numerous companion cells. Sieve tube element and companion cell originate from a common meristematic cell is correct. So here A is false, R is true. Because each sieve element is usually associated with one companion cell is wrong. Each sieve tube element is having its own companion cell. The number of sieve tubes is exactly equal to the number of companion cells. So A is false, R is true. This is the answer for this. Now again a 3-column tabular form. Fiber tracheids, lignified cell walls helps in conduction. Libriform fibers, sclerenchyma, mechanical support. Ray parenchyma, horizontal rows, starch. Tyloses, xylem parenchyma, young vessels. Fiber tracheids show lignified cell walls, but they do not help in conduction. They help in providing mechanical support. Libriform fibers are made up of sclerenchyma. They provide mechanical support is correct. Ray parenchyma, horizontal rows, they helps in the conduction of storage of starch and other things is correct. Tyloses, xylem parenchyma, but not in young vessels. They are formed in old vessels. So the correct combination here is second one. 2 and 3. Wrong combinations are 1 and 4. Regarding tyloses, formed from xylem vessels into parenchyma cells, become lignified at maturity, obstruct the transportation of water and pathogenic fungi, protrude through pit pair. Formed from xylem vessel into parenchyma cell is wrong. Formed from xylem parenchyma into xylem vessels is correct. Here it is given in a reverse order. Formed not from xylem vessels into the parenchyma, but formed from xylem parenchyma and enter the xylem vessels. So first one is wrong. Second, become lignified at maturity. Initially, tyloses are very smooth walled, but later due to the deposition of lignin, they become thick walled. So second, become lignified at maturity is very much correct. Obstruct the transportation of water in pathogenic fungi. This is if secondary xylem vessel. This is if secondary xylem vessel. Suppose it is surrounded by xylem parenchyma. The xylem parenchyma, whenever it ruptures or whenever it gets injured, it produces special balloon shaped structures called tyloses. Special balloon shaped structures called tyloses. These tyloses blocks the movement of water. These tyloses blocks the movement of water. They also prevent the infection of microorganisms. Whenever a xylem vessel shows tyloses, it is said to be very, very durable. It is said to be very, very durable. So answer for this is 2 and 4 are correct. 2 is become lignified at maturity is correct. 4 is protrude through. Second one is correct. Next. Xylem vessels are more efficient in the conduction of water than tracheids. Sap moves from one trachea into neighboring trachea through and bordered pits. Xylem vessels are more efficient in conduction of tracheids is correct. It is proved in almost all plants. Reason? Sap moves from one trachea to neighboring trachea through and bordered pits. So first one is regarding xylem vessels, other one is regarding xylem tracheids. Both are linked to each other, but both are correct. So both A and R are correct. R does not explains the A. Why? Because first assertion statement is given regarding xylem vessels. Reason statement given regarding xylem tracheids. Both are correct but not related to each other. So definitely both A and R are correct. R is not the correct explanation of A. Study the following statements regarding sieve tubes. Identify the incorrect one. Study the following statements regarding sieve tubes. This is belonging to phloem. Identify the incorrect one. They are living with sieve areas highly specialized, localized as sieve plates. Correct. They are living cells with highly specialized sieve areas which show both sieve plates and sieve pores. They are analogous to vessels of xylem. Correct. They are analogous to vessels of xylem means they are structurally similar but function is different. Analogous to the vessels of xylem. How they are analogous? Both in sieve tubes and xylem, the end walls are closed. Vessels of xylem end walls are open. They present as continuous channels from top to bottom or base to apex. Sieve tubes are made up of many sieve tube elements with sieve plate at the one end or end walls or cross walls. It is also correct. Sieve tubes are made up of many sieve tube elements with sieve plates at the end walls or cross walls. Young sieve tube elements are enucleate is wrong. Young sieve tube elements definitely contain nucleus. Mature sieve tube elements do not possess nucleus. Mature sieve tubes are enucleate. Young sieve tubes are not enucleated. So answer is 4. Answer is 4. High quality jute is obtained from. high quality jute. Jute is Carcorus capsularis, which is used in making doormats, gunny bags, tarpaulins and other things. It is normally obtained from secondary phloem fibers. Secondary phloem fibers are also called bast fibers. The method of extraction of jute fibers from its plant is popularly called retting. So, high quality jute fiber is obtained from not from secondary xylem of Hibiscus cannabinus, because Hibiscus cannabinus does not give jute, it gives Madras hemp. Hibiscus cannabinus gives Madras hemp. Secondary phloem of linum. Linum gives flax fiber. Linum gives flax fiber. Secondary phloem of Carcorus capsularis. So, secondary phloem of Carcorus capsularis is correct. Primary phloem of Gossypium herbaceum is wrong. Gossypium herbaceum, the useful part is epidermal hair of the seed coat. Testa of the seed coat becomes cotton, not primary phloem. So, high quality jute fiber is obtained from secondary phloem of bast fibers of Carcorus capsularis. So, answer is third one. Extrafloral nectaries are found in nectary glands. The glands which secrete nectar and attracts the insects to perform pollination. Floral nectaries, extrafloral nectaries. Floral nectaries, extra. Floral nectaries means present on the floral parts. Example, dianthus, cucurbits, hibiscus and many plants you will find floral nectaries. Extrafloral nectaries means on any other part except on floral parts. They are found on the petioles of passiflora. Then they are found on the cup of Cyathium inflorescence in Euphorbia and poinsettia. Cup is formed by the fusion of involucre of bracts. So, extrafloral nectaries are present, 1 on the petioles of passiflora, 2 on the cup-like inflorescence of Cyathium inflorescence. So, here peduncle of passiflora is wrong, pedicel of passiflora is wrong, plumule of passiflora is wrong, but petiole of passiflora is correct. So, extrafloral nectaries are found on the petioles of passiflora, secrete nectar, attracts the insects and help in pollination. This is a matching. Para rubber, simple lattice fur, Indian rubber, lysigenous cavity. This is regarding special tissues and other things. List 2, Nerium, Hevea, Eucalyptus, Ficus, Trapeolum. A is true. Para rubber is obtained from Hevea brasiliensis. Para rubber is obtained from Hevea brasiliensis. Simple lattice fur. Simple lattice fur is Nerium. Simple lattice fur means it is formed from individual cells, may or may not be branched. They are not united to form a complex network-like structure. They are called simple lattice furs. They are found in Nerium. Indian rubber, Ficus elastica. Commercial rubber or Indian rubber is Ficus elastica. Lysigenous cavity is Eucalyptus. Lysigenous cavity is Eucalyptus. So, para rubber, Hevea brasiliensis. Simple lattice fur, Nerium. Indian rubber, Ficus elastica, lysigenous cavity, Eucalyptus. So, answer is 2143. 2143 is the answer for this. Now, another one. Guttation, hydathode, trapeolum, colocasia. Secretion, lysigenous cavity, citrus. Digesting insect, insectivorous plants, digestive glands, Nepenthes. Lattice, laticiferous cells, Nerium. All are correct. Answer for this is 3. Guttation occurs through hydathodes and the examples are trapeolum and colocasia, correct. Secretion is the activity of lysigenous cavity, citrus is correct. Citrus shows lysigenous cavities on its pericarp or epicarp. Dissecting the insects in insectivorous plants because of the presence of digestive glands in Nepenthes. In Nepenthes, digestive glands are present all along the inner margins of the pitcher. So, Nepenthes. Latex secreted from laticiferous shells, independent cells not form a network like structure, Nerium. So, answer is 1, 2, 3, 4. So 3. 3 is the answer for this. Next, match the following items of list 1 with those of list 2. List 1 and list 2. Digestive glands, nectary glands, epithelium, epithem. Hydatodes of pothos, tentacles of drosera, resin ducts of pinus, petioles of passiflora, osmophores of orchids. 1. A2. Digestive glands. They are present at the tip of glandular tentacles in drosera, secrete digestive enzymes which contain proteolytic substances that digest the protein part of insect's body. Nectary glands. Nectary glands is petioles of passiflora, extrafloral nectaries. Epithelium. Epithelium is found in resin ducts of pinus. So secretory cavities are of two types. 1. Schizogenous cavities. The other lysogenous cavities. Schizogenous cavities, lysogenous cavities. Schizogenous cavity means it is formed by the expansion of intercellular spaces. It is formed by the expansion of intercellular spaces, and a special feature about schizogenous cavity is it is internally lined with living epithelium tissue. Living epithelial tissue. The best example is resin ducts in any gymnosperm. Lysogenous cavity. It is formed by the destruction of cells. It is formed by the disintegration of cells or destruction of cells, and here these are lined by dead tissues. These are lined by dead tissue. That is the difference between schizogenous and lysogenous. Lysogenous cavity is internally lined by epithelial tissue. Lysogenous cavity is surrounded by dead tissue. Schizogenous cavity is internally lined by epithelial tissue. Here it is found in epicarp of citrus and leaves of eucalyptus. Leaves of eucalyptus. So digestive, what you call secretory cavities are two types based on their formation. If it is formed by the expansion of intercellular spaces, it is called schizogenous cavity, which is internally lined with epithelium. If it is formed by the disintegration of cell walls, it is called lysogenous cavities. It is internally lined with dead tissue. So here epithelium means resin ducts of pinus. Epithem means hydatodes of pothos. Any hydathode shows loosely arranged or tightly arranged parenchyma tissue called epithem. So digestive glands, petioles of passiflora, nectary glands, digestive glands, tentacles of drosera, nectary glands, petioles of passiflora, epithelium, resin ducts of pinus, epithem, hydathodes of pothos. So answer is A3, B4, A2, B4, C3, D1. This is an assertion reason. Hydathodes are present in the plants where stomata are absent. Hydathodes are present in the plants where stomata are absent. Excess of water in the plant is forced out through hydathodes. Hydathodes are present in the plants where stomata are absent is wrong. Even when stomata are present in the leaves, stem and other parts, hydathodes are present and help in guttation. Stomata will be performing its own function, transpiration. Hydathodes are performing their own function, loss of water in the form of water vapor, that is loss of water in the form of water droplets, that is guttation. So hydathodes are present in the plants where stomata are absent is wrong. Excess of water in the plant is forced out through hydathodes is correct. So here A is false, R is true. A is false, R is true. Next, generally pteridophytes and gymnosperms do not contain. Pteridophytes and gymnosperms do not contain companion cells, sieve tubes, vessels. Xylem vessels in majority of the pteridophytes and gymnosperms absent. Companion cells are absent in pteridophytes and gymnosperms. Sieve tubes are, but sieve cells are present. So answer is 2, 3, 4. In pteridophytes, companion cells are completely absent. Companion cells are totally absent. Whereas in gymnosperms, companion cells are replaced by, they are absent but replaced by special cells called albuminous cells. So, sieve tubes are absent, companion cells are absent, and xylem vessels are absent in pteridophytes and gymnosperms. According to this question, generally pteridophytes and gymnosperms do not contain means companion cells, sieve tubes, vessels. So, answer is 2, 3 and 4. Choose the incorrect combination. Euphorbia, nectaries and latex cells correct. Citrus, nectaries and oil cavities is correct. Nepenthes, nectaries and digestive glands is correct. Cucurbita, nectaries and simple perforation is wrong. Cucurbita contain compound perforation plates. Simple perforation plates are found in whitish. Compound perforation plates are found in cucurbita. So, euphorbia, nectaries and latex cells is correct. It contains both nectary glands and latex cells. Citrus also contain nectary glands at the base of the ovary and a disc-like structure. Oil cavities are definitely present in the epicarp or pericarp of fruit wall. Cucurbita, nectaries and simple perforation plate is wrong. It is multiple or compound perforation plate. Nepenthes, nectaries and digestive glands are correct. Nepenthes shows nectaries on the margins of the pitcher and digestive glands all along the inner walls of the pitcher. So, third option is wrong here. Choose the incorrect combination is cucurbita, nectaries and simple perforation plates. This is another matching. 3-column match. Type of vascular element, special feature, function. Sieve cells, sieve plates present and sugars transport, but in sieve cells, sieve plates are absent. Tracheids, perforation plate, transportation of minerals. In tracheids, you don't find perforation plate. They are found only in xylem trachea where the end walls are closed. Perforation plates are found only in xylem trachea where the end walls are dissolved. Xylem tracheids do not possess perforation plates. Transportation of minerals is correct, but they do not possess perforation plate. Sieve tubes, presence of sieve plate, transportation of sucrose is correct. Sieve tube definitely contains sieve plates and transportation of sucrose. Vessels, perforation plate, transportation of mineral water. Xylem vessels definitely shows perforation plates because the end walls are open. So, definitely shows perforation plate. So, answer is 3 and 4. Answer is 3 and 4. Next, another 3-column matching. Special tissue, distribution in plants, the name of the plant. Digestive glands, pitcher, Drosera. Digestive glands, pitcher, Drosera. Digestive glands are present in pitcher, but pitcher is absent in Drosera. Pitcher is present in Nepenthes. Nectar is petiole passiflora. Nectar is petiole passiflora. Then, osmophores, petals, orchids. Osmophores, petals, orchids. Hydatodes, stem, pothos. So, answer is 2 and 3 here. It is not the third option, it is second option. Nectaries are present on the petioles, passiflora. Hydatodes are present in the stem of pothos is correct. Digestive glands, pitcher, Drosera is wrong. Osmophores, petals, orchids is wrong. Osmophores are not present in petals. They are present in various plant parts. So, third option. Only bordered pits are seen in the following elements of xylem. Bordered pits are the special pits like this. Every bordered pit shows a middle lamellum and the secondary cell wall bends like an arch above this, like this. And every bordered pit at the center shows an expanded middle lamellum or thick middle lamellum called torus. On both sides of the torus, you find thin middle lamellum called margo. So, bordered pits are found only in lower groups like tracheids. They are found only in xylem tracheids and fiber tracheids. Why? Because tracheids are lower group of plants, lower group of xylem vessels or xylem components present in lower group of plants. Xylem truncates are found only in pteridophytes in gymnosperms with bordered pits. Fiber truncates means the non-functional xylem fibers converted into truncates. So, non-functional xylem vessels converted into truncates means they definitely possess bordered pits. So, bordered pits are seen in following elements of xylem means not in xylem parenchyma and xylem truncates, not in xylem vessels. Xylem vessels do not possess bordered pits. Not in libriform fibers because libriform fibers with simple pits. Truncates. Truncates and fiber truncates is the example. Fourth option. Companion cells are absent in. Companion cells are absent in bryophytes. Bryophytes absolutely does not contain phloem. So, thallophyta and bryophyta are popularly called atracheophytes because they do not have vascular tissue, xylem and phloem. In pteridophytes are also companion cells are absent in phloem. In gymnosperms, companion cells are absent but replaced by albuminous cells. So, anyhow, the answer for this where companion cells are totally absent is bryophytes, pteridophytes, gymnosperms. So, answer is 1, 2 and 3. The special tissue responsible for fragrance in flowers. The special tissue responsible for fragrance in flowers is nectaries secrete nectar, sugary sap attracts the insects and help in pollination. Secretory cavities are either formed by the expansion of intercellular spaces as in schizogenous cavities found in resin ducts of gymnosperms or lysogenous cavities formed by the breakdown of cell walls found in the fruit wall of citrus and leaves of eucalyptus. Laticifers are the components which secrete a milky substrate called latex which contain lot of ergastic substances. Rubber is the main source obtained from the latex. So, here the special tissue responsible for fragrance of flowers is osmophores. Osmophores are present in the form of flaps, flagella, cilia and hair-like structures. They emit a very fragrant, beautiful smell. It is found in orchids. So, the special tissue responsible for fragrance of flowers is osmophores. The rubber obtained from the latex of Hevea braziliensis is popularly known as rubber is the product obtained from latex and this rubber is of two types, para rubber, Indian rubber or commercial rubber. Para rubber is used to make small components and commercial rubber is used to make large components, commercial rubber. Indian rubber is a para rubber is obtained from a plant called Hevea braziliensis. Indian rubber is obtained from a plant called Ficus elastica. Ficus elastica. So, para rubber is obtained from Hevea braziliensis. Indian rubber or commercial rubber is obtained from Ficus elastica. So, para rubber here has come from Hevea braziliensis. The question is the rubber obtained from the latex of Hevea braziliensis is known as para rubber. It is not American rubber or British rubber or German rubber. It is popularly called para rubber. Answer is 1. Floral nectaries are seen in. Floral nectaries are seen in. Floral nectaries means the nectary glands which secretes nectar which attracts the insects to perform pollination. They are seen in Euphorbia and citrus. They are not seen in passiflora. Why? In passiflora, nectary glands are present on the petioles. Whenever they are present on any other part of the plant except floral parts, it is called extrafloral nectaries. So, extrafloral nectaries are found in passiflora. If the question is given as extrafloral nectaries are seen in passiflora, answer will be 2. But here the question is floral nectaries are seen in. So, 1 and 3. Euphorbia and citrus are the plants where you will find floral nectaries. Latissiferous vessels are seen in. Latissiferous vessels are seen in 1 and 3. Cannabis and Hevea. Cannabis and Hevea braziliensis shows latissiferous vessels. 3 is the option. 2 and 3. Sorry. Cannabis shows latissiferous cells. Carica and Hevea shows latissiferous vessels. So, answer is 2 and 3. Latissiferous vessels are found in Carica and Hevea. This is a matching. Aloe, Sambucus, Vitis, Olea. Lacunar collenchyma, lamellar collenchyma, compound sieve plates, filiform sclerids and storage parenchyma. Second option. Aloe. 5. Storage parenchyma. Sambucus, lamellar collenchyma. Vitis, compound sieve plates and Olea, filiform sclerids. So, second option. Assertion and reasoning. Vessels provide mechanical strength to the plant. Vessels provide mechanical strength to the plant. Walls of the vessels have lignin. Any xylem tissue show highly lignified cell walls and narrow lumen. Whenever there is a lignin, definitely they give mechanical support. So, vessels provide mechanical strength to the plant is correct. Why they give mechanical strength to the plant? Because walls of the vessels have lignin. So, answer for this both A and R are correct. R explains A. Water stomata are a type of secretory tissue. Assertion. Water stomata are a type of secretory tissue. Any stomata is a secretory tissue. Water stomata is a type of secretory tissue is correct because it helps in the secretion of water in the form of water droplets called guttation. Any stomata is a secretory tissue is wrong. Any stomata is not a secretory tissue. Stomata is a small pore through which loss of water occurs in the form of water vapor called transpiration. So, the answer is A is correct, R is false. Matching. Lysogenous cavity, schizogenous cavity, water stomata, simple lattice for Trapeolum, Euphorbia, Citrus, Drosera, Pinus. A3. Lysogenous cavity is found in citrus. Pericarp of citrus shows lysogenous cavity. Schizogenous cavity is found in Pinus. B5. Water stomata is found in Trapeolum. C1. Simple laticifers are found in Euphorbia. So, lysogenous cavity is found in citrus. Schizogenous cavity is found in Pinus. Water stomata are found in Trapeolum. What are water stomata? Hydatodes. They are found in Trapeolum. Simple laticifers are found in second one, Euphorbia. Assertion. Trichodendron resembles advanced cryptogams in its vasculature. Advanced cryptogams means either pteridophytes or gymnosperms in its vasculature. Why? It contains xylem tracheids in the place of xylem vessel. Trichodendron is a vesselless angiosperm. Trichodendron is a vesselless angiosperm. Because it is a vesselless angiosperm, it resembles the advanced cryptogams in its vasculature. So, A and R are correct. R is the correct explanation of A. So, this answer is 1. In vessels, perforation plates are present in the lateral walls is wrong, transverse walls is wrong, oblique walls is wrong. Usually on the transverse walls, sometimes in the lateral walls is correct. What is a perforation plate? Wherever the end walls are open, if only one plate is there, that is called simple perforation plate. Suppose near the end walls closed, many perforation plates are there, that is called compound perforation plates. So, simple perforation plates, compound perforation plates are found in xylem vessels. They are present sometimes in the lateral walls and sometimes in other walls, usually on the transverse walls, but rarely on the lateral walls. These pteridophytes have vessels. Just now I wrote equisetum and neetum. Equisetum is a pteridophyte with xylem vessels, but neetum is a gymnosperm. Selaginella neetum. Selaginella is a pteridophyte, neetum is a gymnosperm. Selaginella and equisetum. Selaginella and equisetum are the two pteridophytes with xylem vessels. So, Selaginella, equisetum, Drimys and neetum. Drimys is an angiosperm, but neetum is a gymnosperm. So, here Selaginella and equisetum are the two pteridophytes with xylem vessels. In Nepenthes, digestive glands are present on the. In Nepenthes, special feature is the lower portion of the petiole becomes broad, expanded, leaf-like structure, leaf-like phylloid. the upper portion of the petiole is modified into a special tendril. The leaf lamina is modified into a pitcher. The leaf lamina is modified into a pitcher with an opening showing special nectary glands which is slippery in nature. An immovable colorful lid is formed from leaf apex. All along the inner margins of the pitcher you will find nectary glands. Special downwardly bent hair are present on the lower side and also shows an acidic fluid. Insect get attracted towards this lid, sits on the margin because of the presence of nectary glands. It slips into the pitcher. It cannot move out because of downwardly bent hair. It falls into the acidic fluid. The protein part of the insect's body is digested. How? All along the inner margins of the pitcher you will find many digestive glands that secrete digestive juices containing proteolytic enzymes. So, inner surface of the bladder is wrong, tentacles of the lamina is wrong, upper surface of the lamina or leaflet is wrong. Answer is all along the inner surface of the pitcher. The balloon-like structures present in the lumen of vessel are the outgrowths of. Just now I told this is a xylem vessel. On both sides of the xylem vessel you will find living tissue of the xylem called xylem parenchyma. On both sides it is present. Whenever a xylem parenchyma is injured, it starts producing special balloon shaped structures into the xylem vessels which blocks the movement of water, which blocks the infection of microorganisms and other things. So, these special balloon shaped structures which are blocking the movement of water, infection of microorganisms or the other things are called tyloses. These tyloses are balloon like structures present in lumen of vessel is tyloses. Tyloses are the outgrowths of means definitely xylem parenchyma. They are not formed from vessels, they are not formed from tracheids, they are not formed from libriform fibers, but they are basically formed from xylem parenchyma. They are formed from xylem parenchyma. Answer is 1. Vessels are poorly developed in. Vessels means xylem vessels. Xylem vessels are poorly developed in Trichodendron and Remis is wrong because here the vessels are completely absent. Neetum and Selaginella are wrong. These are the two pteridophytes with xylem vessels. Xerophytes and halophytes is wrong because in xerophytes and halophytes xylem vessels are well developed. Xylem vessels are poorly developed means flowering parasites, complete parasites and aquatic plants. In parasites the plants depend on the host and collect the food through hostoria, so no necessary of xylem. In aquatic plant itself lives in the water, water is absorbed by general body surface by diffusion. They also do not require any xylem vessel. So, xylem vessels are poorly developed in flowering parasites and aquatic plants. Answer is 3. Next. Trachary elements present between protoxylem and metaxylem never shows the following thickenings. Trachary elements present in between protoxylem and metaxylem never shows the following thickenings means pitted thickenings. In between protoxylem and metaxylem, protoxylem is a xylem with narrow lumen, metaxylem is a xylem with broad lumen. If they are arranged in protoxylem towards the periphery, metaxylem towards center, it is exarch. It is found in roots. If they are arranged in such a manner that protoxylem towards center, metaxylem towards the periphery, it is endarch, found in stem, and mesarch. So, here the trachary elements present in between protoxylem and metaxylem never show the following thickenings means pitted thickenings are completely absent. Now, morphologically jute is Carchorus capsularis. Morphologically jute is not a xylem fiber of Carchorus, not a pericyclic fiber of Carchorus, not a medullary fiber of Carchorus, but it is a phloem fiber of Carchorus, that is secondary phloem fiber of Carchorus. These are the lessons regarding simple tissues, lessons regarding complex tissues, bits regarding special tissues, and so we discussed all the 4 chapters, that is meristematic tissues, importance of meristematic tissues and their relationships and other things. Permanent tissues, simple tissues, complex tissues, special tissues. Simple tissues are parenchyma, collenchyma, sclerenchyma. Complex tissues are xylem and phloem, and special tissues are many types in the cells.